You're listening to Medscape's In Discussion series on myelofibrosis, a podcast where thought leaders and clinical experts share their diverse insights and practical ideas for optimizing patient care. This series is hosted by Dr. Tania Jain. Dr. Jain is an associate professor of oncology at the Johns Hopkins University School of Medicine, where myeloproliferative neoplasms are the focus of her research. She is also the director of the Adult CAR T-cell therapy program for hematological malignancies at the Sidney Kimmel Comprehensive Cancer Center. Relevant disclosures can be found with the episode show notes on medscape.com or the Medscape app. The topics and discussions are planned, produced, and reviewed independently of advertisers. This podcast is intended only for use healthcare professionals.
Welcome, everyone. We are in our season 2 of Myelofibrosis for Medscape In Discussion podcast. I'm your host, Tania Jain, and we are having an incredible second season of Medscape In Discussion about myelofibrosis. Today, we will talk about endpoints for myelofibrosis clinical trials, and our special guest today is Dr. Ruben Mesa. Welcome, Dr. Mesa.
Thank you so much for having me on the show.
Well, as many of you already know, Dr. Mesa is an international expert in heme malignancies, specializing in myeloproliferative neoplasms. He is also now the president and executive director of Atrium Health Wake Forest Baptist Comprehensive Cancer Center and the vice dean for cancer programs, along with being Charles Spur, MD, professor of internal medicine at Wake Forest University. So here we go, Dr. Mesa. We designed this episode with you to talk about the past and the future of clinical trials in myelofibrosis. So why don't we start with the fact that you have done a lot of pioneering work in quantifying symptoms in myelofibrosis. You generated a symptom score that we continue to use to measure symptom burden and response in terms of symptoms. Tell us a little bit about what inspired you and what was the background of this endeavor when it started.
Well, again, thank you for having me on the show. I would have to give full credit for this effort really to MPN patients. So it was specifically a patient who helped to organize MPN patient meetings, was a very strong patient advocate, patient by the name of Joyce Niblack. Very, very astute lady. She had been a patent attorney. And in the early 2000s, as I interacted with her, she very much pushed to try to collaborate to see how we could better quantify and understand the symptoms that MPN patients faced. She felt, as other patients did, that frequently their external appearance appears normal, yet they can have very significant and debilitating symptoms. And having been actively involved in caring for these patients, I most certainly agreed without question, having heard from many patients that there were difficult symptoms. So, with this, we did the first study which we published in Cancer, which was an online survey of a global MPN community for patients to identify self described symptoms, as well as information about their disease, comorbidities, etc. And with this, it helped establish and validate somewhat of the pattern of symptoms that these patients were facing. Now, soon after this, we started the first trials with the JAK inhibitors, the phase 1 study with ruxolitinib. And prior to this, along with my mentor at the time, Dr. Ayulu Deferi, we had done many trials in MPN patients with thalidomide, with lenalidomide, with interferon, and other agents. And it had been clear that patients typically did not feel any better necessarily with these other therapies. They might have improvement in their anemia, rarely some improvement in their spleen. But it was the JAK inhibitors, particularly the first phase 1 with the ruxolitinib, that demonstrated a dramatic decrease in their symptoms. And this was really notable. And at the time when we started the phase 1, we were not tracking symptoms in any way. So I went to our statisticians at Mayo Clinic who specialize in patient-reported outcomes. There was a very robust biostatistical core for the Mayo Clinic Comprehensive Cancer Center. And I'm like, well, we need a patient-reported outcome form to be able to measure symptoms. So they asked, well, what symptoms do they have? So I described, well, they have fatigue, splenomegaly, symptoms from the spleen, night sweats, fevers, chills, weight loss, headaches. And they start pulling off of the shelf. This was all paper back in those days. They start pulling off the shelf numerous patient-reported outcome forms and said, "Well, we don't really have any good single questionnaire. We have something for itching that we use for burn patients. We have some kind of spleen form for lymphoma. We don't really have anything very good for night sweats. For fatigue, we have something else." And then I asked the question, well, should we develop our own form? And they said, oh my gosh, no. They're like, there's nothing more frustrating than developing your own patient reported outcome form. It's a very arduous process. But we did exactly that, in large part because it was an unmet need. There was really no other good way to do this. So, using that prior information that we had gathered from a global audience of MPN patients, we developed a base set of questions. We validated that with patients and narrowed down to the first iteration, which was the myelofibrosis symptom assessment form. And then validating a patient reported outcome has to be done in the setting of a intervention where, again, you're doing something different. Because when you validate, you're trying to see, well, can the patient reported outcome form measure a difference? Do patients understand the questions? Are the results reproducible? And if there's a change, is the change meaningful? So we validated it through the conduct of the Comfort 1 study, the phase 3 study for myelofibrosis, as well as validated the benefit of response against an anchor which had been already validated with a patient global impression of change. So, in short, it was a process, but it clearly was a huge team based effort
Oh, absolutely.
from patients, clearly investigators. My mentee at the time was instrumental in many parts of this, Dr. Ruben, as well as many international colleagues and domestic colleagues who worked on further validation efforts.
I think it's not just a metric in clinical trials. It has also improved understanding of these symptoms both amongst physicians, investigators, as well as patients. So certainly has helped move this field of myelofibrosis forward, because I can imagine that up until then, a lot of these patients were hearing that these are non-specific symptoms from their physicians just for the lack of recognition of these.
We've learned many lessons from it. The way I describe it, although starting in clinical trials, what you're doing is you are objectifying the subjective. And a kind of a holistic assessment of a patient with MPNs includes their blood counts, includes either physical exam or imaging of their spleen. It may include a bone marrow, and then it helps to quantify their symptoms. And what we found over time is that the symptoms are consistent. Clearly, patients have variable symptoms. Some have them worse than others. But interestingly, as we look at patients globally, the ranking of individual symptoms are very consistent globally. So, for example, fatigue is clearly the number one symptom. And that's true, and we have data in over 40 countries. Now, there's cultural influences in terms of how severe patients will say their symptoms are on a scale of 0 to 10. Now, every patient acts as their own control, but it's interesting, again, the ranking of symptoms is almost universal. If we look at Africa versus Australia versus Uruguay or the US, their order is similar. From my end, they are an insight into one aspect of the biology of the disease. The symptoms are not random, they're not made up, they're not just a surrogate for anxiety or depression. They have a real biological link with the disease.
Absolutely, and it's certainly fascinating. So, moving into the clinical trials piece of this, you've led a number of trials over the years which have led to drug approvals. You've been probably a part of many of these discussions amongst investigators, with the sponsors, with the FDA about inclusion of spleen and symptoms as major endpoints in myelofibrosis clinical trials, and that has quite set the trend. Could you share a little bit about what those discussions look like to include what I would think or what one would think as being relatively unique endpoints compared to, survival and so forth in AML or other studies?
As we think about the impact of a therapy, there are basically two things a therapy can do. Patients can either live longer or they can live better. And everything else that one can measure is a surrogate for one of those two items. Now, compared to AML, part of the challenge in myelofibrosis is that the survivals, although the disease can be fatal, one, the survival is quite variable, and two, it is more intermediate. Patients might live 3 years or 5 years or even 10 years, and in rare circumstances, patients might live 30 years. So, because of that, there is both a lot of literature around assessing myelofibrosis risk to get a sense of how long people live, but also those longer survivals make survival as an endpoint a very challenging endpoint in myelofibrosis. It's a very elegant endpoint in diseases that cause a lot of mortality within a year or two: GBMs, advanced pancreatic cancer, elderly acute leukemia. But in myelofibrosis, it doesn't work very well. So the FDA had evolved to say, boy, we need to be able to measure clinical benefit. Yes, if you could prove survival, great, but was just not a very, very feasible endpoint, particularly in placebo-controlled studies and other things. So how spleen and symptoms evolved was really a quantifiable way of trying to assess patient benefit. Not to say that it's limited to those things. Any individual MF patient might have a number of aspects of disease burden. They may have symptoms. They may have enlargement of the spleen that further worsens symptoms or might harm them in other ways. They may have anemia. They may have the need for transfusions. They may have thrombocytopenia and risk of bleeding. They may have a risk of progression to acute leukemia. So there is some progression-free survival type piece in this as well. But spleen and symptoms, I think, boiled up in terms of a reproducible way of measuring things that made a real impact in terms of how well patients were living, their quality of life, their symptoms, their day-to-day quality of life, not diminishing any of those other aspects. Two, I think they were endpoints that particularly served the efficacy of JAK inhibition well.
Mhm.
I think, again, one prime example, imetelstat has a phase 2 study suggesting a potential survival advantage, but possibly a survival advantage through a mechanism that is different than spleen or symptom improvement. So I don't think that these are the only endpoints that are meaningful, but they've been particularly meaningful for JAK inhibition. And I suspect as we have evolving therapies, we may see other evidence of efficacy that is linked to improvements in length of life or quality of life that become important.
Absolutely agree. And that is probably where we stand now because there certainly appears to be an emphasis from the FDA to go beyond the spleen right now in clinical trials. Now that we have multiple JAK inhibitors, which, as you already alluded to, do a pretty good job in controlling in spleen most of the time. So the bar is now set higher. How do you see clinical trials designed in the future now that are going to be an added therapy to JAK inhibitors, either in front line or subsequently as a subsequent line of therapy?
Well, it's a very good question. As we think about the recent combination phase 3 studies that have had positive data, pelabresib, navitoclax, and others that are coming down the pike. One, we've seen a dramatic, significant rate of spleen response that has been much higher than single agent JAK inhibition. And this may be very important as a surrogate for improvement in survival. It needs to be proven, but there has been a correlation between a spleen response and improvement in survival in particularly long-term data from the ruxolitinib studies. So from my end, I think that that's a very important piece. Now the second piece that is unclear is, do combination studies need to be better than JAK inhibition alone as it relates to improvement in symptoms? They may not be. Again, sometimes with therapy naive patients, JAK inhibition does a pretty significant job of improving symptoms. And it may be difficult for statistical reasons as well as a ceiling effect to be able to show statistical superiority for improvement in symptoms. Now, new agents have other metrics of efficacy that are evolving: improvement in anemia, improvement in the JAK2 variant allele percentage, improvements in fibrosis. Now, these endpoints I think are interesting, but I still view them as somewhat experimental endpoints. That we truly don't know whether on their own they really lead to those two core things I said, which are really the most important at a patient level. Do they correlate with an improvement in survival? They may, but I think we need to prove that. Or two, do they lead to patients living better? Now, if you take a patient who's transfusion dependent and you make them transfusion independent, you probably are making them live better. They have less hassle of medical care, less travel. During COVID, they had less exposure to the healthcare system, which was beneficial. They have improved oxygen carrying capacity and can do their activities of daily living with a greater vigor and less issues. They probably have less risk of heart failure. So I think these other endpoints are very important and will continue to evolve. Things like fibrosis and variant allele frequency, I think we probably have a higher bar to prove. Are these really important in and of themselves, or are they surrogate markers for other things that are important?
I think we certainly have, over the years, the myelofibrosis clinical trial research has been rewarding in a way that therapies to make people live better with myelofibrosis has certainly evolved in the last decade or so, in terms of ruxolitinib, fedratinib, pacritinib, improving spleen and symptoms, momelotinib, hopefully soon luspatercept, hitting some of that transfusion burden that you referred to. Having said that, though, as our good friend Dr. Pemaraju does allude to, transplant still remains the only potential cure. So, how do you see survival becoming a part of drug assessment with the novel drug trials? What does it take to do that? Recognizing, again, to your point earlier, this is a chronic malignancy, and so to speak, a readout of survival can take a long time. So there are challenges to that. How do you see survival becoming a part of myelofibrosis drug trials or clinical trials?
It is a challenge without question. I think we can show improvements in survival in patients that have the shortest survival, which again has some bias that's really baked in with that. We are awaiting the results of the imetelstat phase 3 study, but this was a trial for patients who had relapsed or refractory myelofibrosis to JAK inhibition. And because of that, we know that they have a shorter survival. There's always the potential that an agent might be beneficial earlier in the course of the disease. And when you are testing them only in individuals with a short survival, you may miss the efficacy because the disease no longer can kind of respond because it's a little too far down the pike. I think we still, as a field, one of the greatest unmet needs in myelofibrosis is truly understanding why people progress. Because if we truly knew why people progress, then we would be able to measure that. We would be able to track that. And then we may be able to specifically design therapies really with that in mind. The lack of having such a standard also even really limits us in terms of dose. Do we know, again, if we push the dose of JAK inhibition, are they less likely to progress? There are combination studies that are ongoing, they may vary in terms of their ability to decrease progression-free survival, but we don't have a good surrogate. Even combinations with therapies such as interferon and JAK inhibition, and there's some early data from combinations shown by my friend Jean-Jacques Kilajan. Again, is that the right approach? How much interferon? It remains a key unmet need. Survival will be difficult for us to use as a great endpoint until we get better understanding of mechanisms of progression-free survival and being able to track progression-free survival.
Speaking of interferons, I think that is a great example that has developed significantly over the years and used more and more commonly in early MPNs, like ET, PV, even early myelofibrosis. And now, as Dr. Kalajian's work now evolves to show that there may be some benefit in progression-free survival with JAK2 variant allele fraction reduction. Something along those lines in the context of myelofibrosis is certainly awaited. I appreciate your time, Dr. Mesa. We're hitting the top of our time. It was an honor to have you, especially since I guess I say this often, but you're probably one of the big reasons why I do what I do. You kind of got me into the MPN field. So it was an honor having you and learning from you. Would love to hear more from you in the future.
Well, thank you again so much for having me on this show. And again, I've been following your episodes with great interest.
Thank you, Dr. Mesa. That means a lot.
Thanks for listening to Medscape's In Discussion Myelofibrosis podcast series with our host, Dr. Tania Jain. Be sure to look for more In Discussion episodes wherever you get your podcasts. Check out medscape.com or the Medscape app for show notes, links, and more information on myelofibrosis.